Hello class and welcome to Module 12. Our topic for today is going to be avoidance and acclimation to freezing stress. To start off this lecture, we'll discuss acclimation and see how it happens in nature. Then we'll look at the changes that accompany acclimation, after which we'll discuss the following changes in details: gene expression, proteins, sugars, and membrane adjustments. After that, we'll look at some plant freeze protection practices. And lastly, we'll shed some light on cryopreservation. In this module, we'll be dealing with freeze-tolerant plants. Even though these plants are freeze-tolerant, ice must be formed extracellularly. Intracellular freezing is lethal. The figure shows extracellular freezing of leaf mesophyll cells. The control cells are unfrozen and turgid. The frozen tissue shows large extracellular ice masses. Confirmation of extracellular freezing is provided by removing the ice through sublimation. There are large voids where the ice was, and cells are shrunken to a small fraction of their original volume. Freeze-tolerant plants can undergo a transition from a freeze-susceptible to a freeze-tolerant state. Acclimation is the process of developing resistance to freezing. Once acclimated, the killing temperature is lower than the freezing temperature. Some plants can only acclimate a few degrees, while others can survive immersion in liquid nitrogen under the proper conditions. Plant tissues such as seeds have survived exposure to temperature approaching absolute zero, indicating the capacity to survive any low temperature exposure expected on Earth. Acclimation can be triggered by several factors. Changing photoperiod. Longer nights bring about the first stage of acclimation in many woody plants. The leaves are the receptors of the phytochrome-mediated response. The second stage is induced by low temperatures. Herbaceous perennials generally respond to temperature only. A mild water stress can also result in a moderate increase in hardiness that is not cumulative with other triggers. As you recall from previous modules, many biochemical and physical changes take place during acclimation. There is also compelling evidence that certain genes are induced during acclimation based on production of new mRNA and proteins. Tissue water content decreases, cell wall and protoplasm can be augmented, membrane composition can change, including greater unsaturation, and soluble sugars generally increase. In this module, we'll discuss some of these points in more details. While there are still many unknowns, the picture is developing concerning signal transduction and regulation of gene expression during low-temperature acclimation. The primary temperature sensor is not known with certainty, but evidence suggests changes in membrane fluidity are involved. In lower organisms, membrane rigidification activates a mechanosensitive histidine kinase similar to the two-component system. Cytoskeletal involvement is also likely. Increased cytoplasmic calcium may interact with calmodulin or calmodulin-like proteins. Evidence is accumulating that H2O2 also acts as a second messenger. A likely site of production focuses on complex 1, a dehydrogenase in the mitochondrial electron transport pathway that is altered by low temperature. Then comes the phosphorylation state. Protein kinase activity increases involve both calcium-dependent protein kinases and mitogen-activated kinase cascades, possibly corresponding with each of the second messengers. Phosphatase activity decreases. And then lastly, gene induction involves both cis and trans-acting factors. Several batteries of genes involved in cold acclimation have been identified. However, the best studied group is the CBF regulon. The CBF pathway is a transcription factor cascade. Low temperature triggers activation of genes encoding the CBF transcription factors which interact with cis elements of the CBF regulon. The resulting proteins lead to increased freeze tolerance. As we look at the regulation of CBF transcription, a key question is how do low temperatures trigger production of the CBF transcription factors? Clearly, there is a constitutively produced factor that is inactive in the absence of low temperature signal. The constitutively expressed factor is ICE1, inducer of CBF expression. It's an MIC basic helix loop helix transcription factor. Once activated, ICE1 triggers CBF3 transcription by binding to ICE-R1, induction of CBF expression region, which is also an MIC cis element in the CBF3 promoter. It is assumed that similar transcription factors exist for other CBF genes. The cis element of the CBF promoter is known by several acronyms: CRT, which stands for C-repeat, DRE, which stands for dehydration responsive element, and LTRE, which stands for low temperature response element. The trans-acting factors belong to the epitella to ethylene response factor superfamily of transcription factors. CBF stands for C-repeat binding factor. CBFs are also known as DREBs, dehydration response elements binding proteins, and therefore belong to the DREB family. CBF DREB1s are triggered by low temperature, and DREB2 is induced by dehydration and salt stress. We will now shift our attention from regulation of freeze acclimation to the function of the gene products. The proteins produced during cold acclimation are known by many names, partially due to having many different labs working in the area. Core proteins or cold regulated proteins are probably the most studied. Characteristics of many of the core proteins have been described. While we normally think of proteins unfolding and aggregating at elevated temperatures, some core proteins are boiling stable. They tend to contain a high proportion of hydrophilic residues and are comprised from relatively few amino acids with repeating motifs. Core proteins are also known for a tendency to form a large proportion of alpha helices. A number of studies sequenced freeze-induced proteins and checked for homology with known proteins. Matches came up in a number of areas, most of which made sense based on what is known about injury mechanisms and acclimation pathways. LEA proteins, which stand for late embryogenesis abundant, are boiling stable proteins. They are produced during seed formation just before drying. LEA proteins are thought to be involved in resistance of developing embryos to water stress, and their action may be related to their hydrophilic properties. AFPs are antifreeze proteins. Those help enable certain insects and polar fish to survive low temperature. PR proteins or pathogenesis-related proteins such as chitinase, thaumatin, and glucanase are involved in disease resistance. However, it is hard to separate functions since some chitinases have antifreeze proteins activity. Some other proteins that showed sequence homology are lipid transfer proteins, proteases, catalases, and protein kinases involved in transcription factors. Now let's look at the freeze-induced proteins' functions. Some of these have antioxidant roles. While we may not immediately think of oxidative reactions being associated with freeze damage, evidence of involvement includes depleted antioxidants and lipid peroxidation in freeze-damaged tissues. McCarthy has suggested that leaks from electron transport chains are one source contributing to activated oxygen species. Ice tends to exclude solutes and gases, leading to a high local partial pressure of oxygen, which favors oxygen reduction, especially in the presence of a reduced metal ion. Acclimated plants were more tolerant to superoxide, and transgenic plants that are overexpressing SOD had greater freeze tolerance. The second role is regulatory. Regulatory roles are evident in proteins such as transcription factors. Molecular chaperones can protect against freeze-induced denaturation. The third role is antifreeze or thermal hysteresis. Antifreeze or thermal hysteresis proteins have interesting roles in response to stress. Polar fish were among the first organisms identified to utilize antifreeze proteins for freeze protection. Scientists working with insects refer to them as thermal hysteresis proteins. Antifreeze proteins have dual roles. They function to inhibit crystal growth and they inhibit recrystallization by preventing small ice crystal to condense into a larger one. Recent work has also shown that antifreeze proteins may help stabilize membranes at low temperatures via binding. Some proteins may act directly to protect enzymes and membranes, like Core15. Core15 is one member of the CBF regulon and is thought to reduce the incidence of HEX2 formation by binding to the inner chloroplast membrane and changing the curvature of the membrane. This is significant because the likelihood of forming HEX2 phase is related to the curvature of the membrane. The figure shown on the right shows the mode of action model of Core15 protein function. The cellular dehydration induced by freezing results in crowding effects, triggering partial folding and association of Core15 proteins with the chloroplast's inner envelope membrane. This association in turn enhances the helical conformation of the Core15 proteins. Protein binding stabilizes the inner envelope membrane against the formation of hexagonal two-phase lipid domains involving adjacent membranes such as chloroplast outer envelope and the plasma membrane. This finally results in maintenance of plasma membrane integrity during freezing, which can be proved by the reduced electrolyte leakage. Sugars play several roles in acclimation to freezing. They osmotically reduce freeze-induced dehydration by lowering the water potential gradient to extracellular ice. Some sugars can also play the functional role of solvent to help stabilize molecules when water content decreases drastically. Sugars can act as electron donors and scavenge activated oxygen. Sugars aid in the formation of glass transitions. The metastable state formed from highly viscous solutions. They take on properties of an amorphous solid, stopping all diffusion-controlled reaction and processes, including freezing. Cells with glass transitions are essentially in a state of suspended animation. Glass transitions are favored by certain sugars, such as trehalose, and require dehydration leading to supersaturation. The secondary sugars are thought to prevent reaching an eutectic point where the supersaturated solution forms a precipitate. Acclimation involves changes in membrane metabolism that alter membrane properties. Proteins that alter lipid metabolism include acetyl-CoA carboxylase, which catalyzes the first step in the fatty acid synthesis, desaturasses that add double bonds to maintain fluidity at lower temperatures, and phospholipid transfer proteins that have roles in trafficking between organelles. One of these shifts as plants acclimate is to increase the proportion of glycerol-based phospholipids such as phosphatidylcholine or PC and phosphatidylethanolamin PE at the expense of cerebrosides. Some scientists believe that increased levels of phosphatidylcholine with unsaturated fatty acids are largely responsible for avoiding dysfunctional membrane configurations like the HEX2 phase. The second membrane adjustment during freeze tolerance is membrane resealing. To understand this phenomenon better, we should first learn more about SYT1. SYT1 belongs to a small gene family, and the Arabidopsis genome contains 5 genes encoding synaptotagmin-like proteins SYT1, SYT2, 3, 4, and 5. Those proteins act as calcium sensors. When the plasma membrane is mechanically punctured by ice crystals, calcium influx occurs from the extracellular space into the cytoplasm through the damaged site. The C2A and C2B transmembrane domains are two tandem calcium binding domains that attract calcium. Synaptotagmins interact with membranes composed of negatively charged phospholipids as C2A and C2B domains. This triggers calcium-dependent exocytosis, which is the process in which intracellular vesicles move to the plasma membrane and subsequently fuse with it. Endomembranes then fuse at the site of the damaged plasma membrane via calcium binding SYT1, and consequently the damaged site is resealed. The third membrane adjustment is the formation of freeze-induced vesicular structures. The formation of many vesicular structures had been observed in isolated protoplasts of cold acclimated Arabidopsis leaves. They appear in the cytoplasm near the plasma membrane right after extracellular freezing occurs. Those structures are referred to as freeze-induced vesicular structures, or FIVs. When FIV fuses with the cell membrane, they release the mechanical stress that is exerted upon the membrane during freezing. In other words, cold acclimated plants may mitigate tension in plasma membrane by regulating its surface area. Another example on this is Fujikawa and Takabe findings in mulberry cortical parenchyma cells. These cells acquired extreme freezing tolerance by formation of multiplex lamella, or MPL, by the fusion of ER vesicles. This is shown in the image in front of you. This table summarizes all the injuries that can occur in a cell membrane and also shows how tolerant plants overcame those injuries. On the first row, we can see the hex 2 phase injury, which was overcome by the increase of unsaturated species of phosphatidylcholine. In the second row, we can see the fracture jump lesion, which was overcome by increase in hydrophilic proteins and compatible solutes. Another way woody plants have overcome fracture jump lesions was by increasing in ER vesicles. Next, we can see that the mechanically disrupted membrane by ice can be overcome by increasing the SYT1 component, which leads to membrane resealing. In the last row, we can see that expansion-induced slices in protoplasts was overcome by increased unsaturated species of phosphatidylcholine. For centuries, growers have attempted to protect susceptible plants from freezing and to reduce damage to freeze-tolerant plants. There are many terms related to freeze protection, some of which have variations depending on meteorological conditions. It is common to distinguish between light frost, which is frequently associated with some damage to susceptible plants, and a hard freeze that is lethal to plants that must supercool to survive. Other people make the distinction based on the modes of thermal energy transfer. Radiation frost can occur when there is little or no wind with a clear sky. Plant canopies lose heat by black body radiation under these conditions. The outermost leaves can be colder than air temperature and freeze while the rest of the plant survives. Advective freezes, which occur from movement and mixing of air, are commonly associated with hard freezes. However, the distinction between types is somewhat arbitrary because thermal energy transfer can involve several modes. Freeze protection practices can be categorized into direct and indirect methods. The indirect methods of freeze protection are usually passive, requiring little or no inputs at the time of the freeze. Plant material with sufficient capacity to acclimate can be chosen. Avoiding low spots where denser, cold air drains and avoiding locations behind restrictions to airflow can reduce the probability of damage. Unconventional wisdom states that it is best to avoid cultural practices that promote growth flushes late in the season, such as heavy pruning or fertilization. The direct methods of freeze protection usually involves activity at or near the time of low temperature episodes. Many are based on microclimate modifications, protection based on preventing low temperature exposure in the site of the injury. The first one are covers, which prevent loss of outward radiation. But individual plant covers such as cups, heat caps, and clutches have been used. They tend to be labor intensive and not well suited for large plants. The second direct method is irrigation. Irrigation for freeze protection is very common and involves several approaches. Flooding provides a few degrees of protection during radiative freezes. Effects are due to sensible heat, water's high heat capacity, and warming from water condensing on the plant. Sprinklers irrigate the crops so that water is continually freezing. While water is freezing, it remains at or near the 0 Celsius degree. Since the tissue solution contains solutes, its freezing temperature will be slightly below 0 degrees Celsius. Therefore, the plant will not freeze even though it's encased in ice. Problems with this approach include breaking the plant from the weight of the ice and evaporation. The method is only beneficial when it is very calm because more heat can be lost from the evaporative cooling than gained from the heat of fusion. Creating fog can block outward radiation of heat. Problems with fog include having to blow away or evaporate. The third direct method is heaters. Heaters have been used for centuries, but the cost has become prohibitive except for high-value crops. Perennial plants may warrant the expense in some instances. Pollution in this case was another drawback, because in the old days they burnt everything from coal to old tires. The fourth direct method involves mixing air. A temperature inversion frequently forms under radiative conditions. The air at ground level is colder than the air away from the ground in inversion situations. Wind machine and helicopters can mix the air, raising temperatures near the ground. The fifth method is limiting heterogeneous ice nucleators, which is a novel method of freeze protection involving limiting ice nucleation. Rather than trying to keep the plant warm, this approach is aimed to maximizing supercooling. Approaches to increase supercooling are primarily based on the premise that ice-nucleating activity bacteria limit plant supercooling. Several methods of controlling populations of ice-nucleating activity bacteria and/or their capacity to trigger freezing have been proposed. Antibiotics are more of a theoretical interest due to problems associated with the selection of resistant strains. Scientists have reduced populations of INA bacteria with antibiotic sprays, but reports of reduced freeze damage have not been consistently repeated by others. Nucleation inhibitors are compounds that reduce ice nucleation activity without necessarily killing the bacterial cells. Some may work by reducing protein aggregate size. The problem that arose is that those compounds capable of disrupting ice-nucleating proteins were also phytotoxic. Sprays containing antitransparents and clay particle have been tried with limited success. Biological control involves the introduction of competitive bacteria that are not ice-nucleating active. The idea is for the competitive bacteria to use the available nutrients and delay or reduce colonization by INA bacteria. This approach is where genetically engineered organisms came in, although naturally selected bacteria could also be used. Lindo, a scientist at Berkeley, developed an ice-minus bacteria for freeze protection. As the story goes, court battles delayed testing this genetically modified organism, then demonstrators destroyed the research plots. A company was also working on the approach and was giving the go-ahead for field testing. This was back in the time of the Ghostbusters movie. AGS representatives dubbed themselves Forest Busters and called in the media to film the historic event. When folks watching the news saw scientists in full contaminant suits, some may have wondered just how dangerous this stuff was. The commodity groups pulled their support and the approach pretty much went away. As we started recognizing the need to maintain genetic diversity, cryopreservation was the way to go. Cryopreservation is a method being explored for maintenance of germplasm for extended periods of storage at very low temperatures. It is very expensive to maintain plants and germplasm collections in orchards, where they are susceptible to loss from diseases, insects, and natural disasters. Germplasm repositories are supplementing growing plant collections with cryopreservation. In addition to this important function, cryopreservation has provided some valuable clues to how plants survive exposure to very low temperatures. A number of the protocols are aimed at achieving glass transitions. A number of factors have a bearing on whether plant tissues can be stored successfully at low temperatures or not. Advantage of plant-based mechanisms can be exploited using naturally acclimated tissues. The normal reduction of water content during natural acclimation can be augmented. Scientists have had success with approaches using controlled rates of temperature change and the addition of cryoprotective agents. Cryoprotectants such as glycerol can penetrate cells while others are non-penetrating. Several cryopreservation protocols have been developed. Slow cooling is a two-step process. Nucleated samples treated with a cryoprotectant such as dimethyl sulfoxide are cooled at about 1 degree Celsius per minute to about minus 40 degrees Celsius. Cellular dehydration from extracellular freezing plus high concentrations of the cryoprotectant enable the tissue to undergo a glass transition upon being plunged into liquid nitrogen. Warming is usually very rapid. One protocol using rapid cooling rates is termed as encapsulation. Tissues are encapsulated in alginate beads treated with high concentrations of sucrose, dried then plunged into liquid nitrogen. Another simpler procedure involves treating tissues with a cryoprotecting cocktail, both penetrating and non-penetrating compounds followed by liquid nitrogen. Non-differentiated tissues like suspension cultures and callus are mainly preserved by slow cooling protocol. Meristems and embryos are generally suited for the rapid cooling procedures. However, the source is also a factor, with hardwood shoot tips working well with rapid cooling, but softwood rely on slow cooling of non-differentiated cells. While it is common practice to store seeds in conventional freezers, which is minus 15 to minus 20 degrees Celsius, the period of viability can be greatly increased by storage at much lower temperatures. At the end of this lecture, you should know the answers to the following questions. How does acclimation help plants survive freezing? How do plants acclimate in nature? Name the physiological changes that occur in freezing acclimated plants. And what is the major regulon controlling cold acclimation? What are the characteristics of core proteins and what are their roles? How do sugars help in acclimation to cold? Name the 3 adjustments that plasma membrane undergo in freeze acclimation. And what is freeze protection and what are its types? And what is cryopreservation and what is its purpose and how is it done? Thank you and we'll see you in the last module.